Hi, everyone. Today we are going to start talking about the role of the immune system in protecting certain surfaces of our body that we have only sort of mentioned before, but we're going to take a look at in more depth. And that has to do with the mucosal layers in our anatomy. And we will discuss, what is the importance of the mucosal layer? Why is it such a big deal when we are thinking about our immune system? And also talk about how our immune system has to have specialized certain types of responses and features for protecting the mucosal layers of our body. When we think about physical barriers are the first kind of defense against microbial infection. Right? Whether you're talking about viruses or bacteria or parasites, we rely upon physically keeping them out. And so when we think about our skin, for example, throughout the semester, as we've been studying things like our immediate innate responses and induced responses, and then the involvement of our adaptive responses, we oftentimes have used examples of things like, oh, you get a cut in your skin and some bacteria get into the tissues of your arm, right? We use that as sort of the starting point because it's a nice way to introduce concepts of innate immunity and then adaptive immunity. As we have moved on to talking about adaptive immunity, B cells and T cells, you notice that we were talking more about situations where pathogens enter our body's tissues by means of things like inhaling them, right? Our respiratory tract or our GI tract when we swallow things. So this makes us realize that even though the most obvious physical barrier that we have to keep pathogens out is our outer skin, right? It's actually not the most important barrier. So you think about this, right? In our everyday lives, when we go out and we're in our different environments, whether at home or work or school or, maybe hanging out like at the beach, right? We are surrounded by microbes. And that's why we rely upon having our thick, tough outer epidermal layer to protect us. Right? But let's expand our discussion about different surfaces of our body where microbes threaten to enter our tissues. The skin and our mucosal layers are very different. Let's talk about our skin first, because that's the easier one to kind of intuitively understand. Right? Our skin, our outer skin, our epidermis is made up of a tough, impenetrable layer or many layers of keratin, right? That comes from the keratinocyte cells that are in our dermal layer, and they make keratin protein. And then after those keratinocytes die, right, they leave the keratin behind. And we have many, many layers of keratin that provide a tough, impenetrable layer. Okay? I think you're all aware that it's pretty tough and impenetrable, even for things like water, right? It takes a long time of you soaking your hand, or your skin in water before you really begin to see any sign that the water is soaking in, right? Like if you're swimming at a long time and then you get really wrinkly, that takes place after like maybe, an hour or 2 hours of soaking in water, right? So if it's that sort of difficult for even water to get through our keratin, you can imagine it's really hard for things like microbes, like bacteria, viruses to come in. Right? And that's why we evolved that way, to have that. Right? In order for us to be able to do any kind of exchanges from inside of our body to the outside in terms of things like different things like sweat, for example, right? We have to have glands that are in our skin that take care of actually providing a way for certain things to be released from our body through the skin. Right? And that's why we've used the skin as sort of our first starting example of our innate immune system way at the beginning of the semester. We said that underneath your tough, impenetrable keratin layer, you have your living cells, the keratinocytes, and then within that tissue, you have your things like macrophages and dendritic cells, and also complement proteins, right, that are in there. Okay. So all of that we've been talking about in terms of how our innate effectors respond in something like our arm, we can now kind of give that a new sort of category, right? We'll call that our systemic immune system, right? In other words, the immune system that deals with throughout the outside of our body, right? That's how we have these innate responses. So a good example, of course, is our cutaneous membrane, our skin. What are our mucosal layers? Right? Mucosa is basically all the stuff that lines these different types of tracts throughout your body, like your respiratory tract, right? From the inside of your nose, down your trachea, down into your lungs, or your GI tract, right? From your mouth through your esophagus, stomach, and intestines. Right? Those are lined with a very different type of tissue. And I think you all intuitively realize this, right? Is that lining is not tough and impenetrable. It's thin, it's accessible tissue because it has to be in order for those different surfaces in these different tracts in our body to actually be able to do their job. When you breathe in, there has to be gas exchange between the air in your lungs and your blood, the blood that's flowing in blood vessels surrounding the alveoli of your lungs, right? So that obviously can't be a thick, impenetrable layer of skin, right? It has to be way thinner, right? And it's very delicate to allow for gas exchange. Same thing with food, right? As we eat food and we digest food, first in our stomach and then our small intestines, if we want to be able to absorb nutrients from the food that we are digesting, it certainly cannot be that the lining of our intestines is thick, impenetrable skin, right? It has to be delicate and thin and accessible. Right? Therefore, that poses a whole set of new problems, right? Because we're talking about membranes that form the boundary between inside of our body and outside of our body, right? That are, unfortunately, by design, have to be very thin, very delicate, and therefore much easier for things like microbes to cross. So mucosa is the main entry point for many microbes. So I ask you to remember the definition of outside versus inside that we use as immunologists, right? So I always said, when you're a little kid, ever since you're a little, toddler, you always had this impression of when mom and dad's feeding you some mashed up peas on a spoon, right? It's outside. And then once it gets in your mouth and they make you swallow it. Oh, now it's inside my body, right? But I said, now as immunologists, we have to think about it differently, right? Because food that's moving down from our mouth into our GI tract, on the esophagus, stomach, small intestine, large intestine, and then finally waste products that are released from our bodies, right? All of that is just passing through our bodies, right? Just like the example I gave you of a car passing through a tunnel in a mountain, right? The car is never in the mountain, it is passing through the mountain, and it depends upon the concrete barrier of the tunnel, right, to separate inside and outside of the mountain. So mucosa then, right, is the lining that really defines inside versus outside for all of these types of systems inside of our body. So our GI tract, our respiratory tract, the urogenital tract, even things like mammary glands, right, are all examples of thin mucosal barriers between inside of tissues of our body and the outside. Think about this also. Those types of places, your mouth, your GI tract, your lungs, right? Those places are potentially filled with microbes. It is a wonderful environment for certain microbes to exist because they are protected, they're provided nutrients, they're provided moisture, right? And so it wouldn't be surprising that, hey, if you know that on the outside of your arm, there's going to be lots of bacteria just because, it's exposed to the outside world, right? You don't think about it because you cannot see the bacteria. Suddenly, if you think about, wow, what about inside, right? My GI tract, right? Certainly that lining is also teeming with bacteria, right? And I want you to think about this, right? compared to the relatively inhospitable, outside of your skin, because the inside of your GI tract, right, for example, is so much nicer for bacteria to grow in. It turns out that there is a ton more microbial contact between pathogens and your body's, potentially your body's tissues, taking place on these mucosal surfaces, right, compared to just what you think about on the outside of your skin. And there is an incredible amount of mucosal surface area, right, compared to the outer skin that we have. So this cartoon just reminds us, right, all of these are examples. Whether you're talking about your respiratory tract, GI tract, here's your urogenital tract, right, here are the mammary glands. These are just examples of all of these feature mucosal layers that separate the interior tissues of the body with the outside world. So how much surface area are we really talking about here to help you appreciate this? So it's kind of shocking because you typically think about surface area as being the outside of your body. You don't think about the surface area of these membranes inside of your body, right? Or at least I should say that you don't see from the outside. Think about it this way. I'm like 6 feet tall, and if I were to just kind of like imagine unwrapping all the outer skin from my body and figuring out how much surface area I'm dealing with, that might be like, for example, the surface area of this picnic table here, right? So if this picnic table is roughly 3 feet wide by 6 feet long, 3 times 6, that's 18 square feet, right, of outer skin surface area. What about if we were to take all the surfaces of my GI tract, respiratory tract, urogenital tract, etc., right, and unwrap that and spread it out? How much surface area would that be? I think you can already guess just by looking at that picture. That would be about 250 square feet, right, of surface area. So about the size of this tennis court, right? And when you consider, like I said before, right, that surface area, right, is actually much more conducive for things like microbes to be happy living on, now you see, right, that there is a tremendous challenge here that our immune system faces when it comes to dealing with, right, so much surface area of our body being exposed to so many microbes. Okay. So we call these surfaces mucosal layers because they are coated with a thin layer of mucus, right? And that mucus helps keep these fragile, delicate layers lubricated, hydrated. They help protect the pH environment of the epithelium, especially in places like your stomach, right? Places like your stomach that I'm sure you're all aware, right, is a very, very extreme environment. The pH is very low. Like pH can be 1 or 2 due to the hydrochloric acid that your stomach is pumping into it, right? Have you ever thought about the fact that, why is it that our stomach doesn't actually digest itself? After all, let's say you're eating a piece of steak or something like that, right? And you have a nice piece of steak and you swallow it, you expect your stomach acid to help break it down. But is that piece of steak all that much different than the lining of your stomach itself, right? So your stomach lining is protected by a layer of mucus, and that mucus is very effective in neutralizing the pH. Mucus also allows for the exchange of molecules. Different things can go back and forth between the outside of our body and into our tissues through first passing through mucus. And of course, when we're talking about the immune system, mucus is a very effective barrier against pathogens, right? Not only providing a block that blocks microbes from getting into your tissues, but also mucus is a nice way to conveniently eliminate things like pathogens. So as you breathe in, with every breath you take, right, there's dust particles, dirt particles, and yeah, you better believe it, right, fungal spores, airborne, viral particles, whatever, right? If you breathe these down into your system, into your lungs, if you didn't have a way to kind of get them back out again, right, you'd basically be collecting all this trash and garbage and microbes in your lungs, right? So the mucosal lining of our respiratory tract which is pictured here in this cartoon. This is a very 70s kind of cartoon, but I like it, because it gets the point across. You would have, for example, in your nasal epithelium, right? The lining of your nose, is already it's moist, it's delicate, it's very sensitive on the inside. And that's because the epithelium is very thin. So in some types of epithelial layers, the outer surface of cells is not more than just 1 or 2 cells thick, right? That's crazy thin if you're talking about the actual layer of cells is just 1 or 2 cells, right? So these cells have on their outside, they're protected by a layer of mucus, right? So this could be a fraction of a millimeter thick of mucus, or in some places in our body, it could be up to like, half a millimeter or more of mucus, right? For example, in places like our stomach or our small intestines. So that's where things that you breathe in would get trapped. So here's virus particles, bacteria, and many of these epithelial cells are specialized. They have things like cilia, right? And these cilia, they beat in one direction and they actually cause the mucus to kind of flow. So the lining of your respiratory tract, right? All the cilia on these cells are constantly pushing the mucus up so that all the stuff that you've breathed in can eventually be expelled a lot easier, right? And that's why, you cough and you sneeze to expel a lot of these particles. All right, what is mucus? Right. Right. Yay, mucus. Right. So, we all know on one hand what mucus is, right? Because our whole lives we've dealt with mucus. But, what is the composition of mucus? So, mucus is mainly water, all right, with certain types of proteins in it, proteins that make it slippery and, mucusy, right? The main protein that's in mucus is mucin. And so mucin is this massive glycoprotein. What is a glycoprotein? Remember, all proteins are chains of amino acids, right? Some proteins fold up into a 3-dimensional shape. Other proteins can actually function as a long chain, a long polypeptide chain. And in this case, this mucin polypeptide chain, right, has lots and lots of short little sugar groups, oligosaccharides attached to them. So here in this cartoon, here the pink part is the actual protein part, or I should say the actual polypeptide chain, because you zoomed in, you're like, oh, what are those? Oh, I know what those are. Those are amino acids, right, in here. But then all these feathery looking things attached to them, zoomed in here, are chains of sugars, right? So these are what we call O-glycans. So these are sugar chains that are attached to, for example, serine or threonine amino acids in here. So as a result, it gives this glycoprotein a very feathery-like property, right? So here are 3 strands of them together, and here's a whole bunch of strands of them together. So this makes it very effective as a lubricant, right? Because the sugar molecules, they can slide very easily over each other. So that's what makes mucus very kind of slick, right? And it also provides a meshwork or a network where things can be trapped, right? Like particles, for example, or pathogens. And this immune mucus, or I should say this mucus, right, is filled with things like immune cells. So you actually have immune cells that occupy the mucus, and components, things like antibodies, right, are part of your mucus as well. So the challenges then, when we stop and consider this, are that the mucosal layers are designed to, or they have to be designed in order to balance 2 very diametric or opposing types of considerations. On the one hand, mucosal layers have to be thin, they have to be delicate, right, in order to allow them to function. So stuff moving back and forth between them, whether you're talking about gases or nutrients or waste products, whatever, right? But then at the same time, as I mentioned before, pressed up against these mucosal layers, right, are massive amounts or massive numbers of pathogens like bacteria, viral particles, other types of potential antigenic types of signals that come from the outside. So how can it do that, right? How does the immune system have to deal with these 2 separate kind of challenges here? Right? So let's look at this. So here's our gastrointestinal system from mouth through the stomach, intestines, eventually out the anus, right? So this is the tunnel through the mountain, right? And food that passes through here from one end to the other, right? Most of that food is never really in your body, but as it gets digested, nutrients and things, right, that are broken down get absorbed into your body through the mucosal layer. About 3/4 of the body's lymphocytes and plasma cells are found in mucosal lymphoid tissues, right? So wow, right? We've been talking about B cells and T cells in terms of going between your blood and your lymph, right, as where they normally do their jobs, right? But about 3/4 of them at any given time have actually settled in to these different mucosal layers of your body to protect you. Also, about 3/4 of all antibodies made at a given time are actually dimeric IgA subclass, right? And they are found in mucosal surfaces. So you might say, wait a minute, Dr. Shen, I thought you told us before that like the most common, antibody that's floating around in your blood is IgG, right? Yeah, that's the most common type of antibody that's floating in your blood, right? So about 70% of all immunoglobulins or antibodies that you find floating around your blood are IgG type, right? But overall, 3/4 of all antibodies that you have in your body right now, right, are IgA that are in the mucosal layers protecting you right now. Protecting you against what? Think about our GI tract. If we were to think about what kind of microbial populations there are in our GI tract, let's start with the mouth, right? In your mouth alone, there are about 750 different species of bacteria, right, that are commonly found in your mouth. If you move down into your stomach, the concentration of this bacteria is typically around 1,000 individual bacterial pathogens per milliliter of stomach fluid, right? That's a lot, right? It gets even worse, right? By the time you get down to your small intestines, because obviously your stomach acid can in fact destroy a lot of the bacteria, right? But by the time you get down into your small intestines and the pH of your stomach fluid has been neutralized by things like bile and things like that, right? Your small intestines, right, is an amazing place for bacteria to grow. So back down there, you have about 10 to the 5 to 10 to the 8 bacterial particles per ml. And finally, in your large intestines, the colon, you have something like 10 to the 12 bacteria per ml, right? That is a lot of bacteria, right? So it turns out that, I know this sounds really pleasant, but the bacteria that you expel from your bodies as part of solid wastes comprise about 1/3 of the solid material, right, in fecal material. Right? So that's how much bacteria, right, you're actually kind of growing in your body at a given time, right, as evidenced by how much you're actually, kind of expelling, right? Boy, it's good that we had this after lunch, this discussion, rather than before lunch, right? So all joking aside, though, I want you to appreciate, right? Yeah, we're talking about a lot of pathogens, potential pathogens, pressed up against a massive amount of surface area, right? That represents the boundary between outside and inside of our tissues. And on top of that, that surface area has to be thin, delicate mucosal layers, right? So what a bunch of challenges. Let's look at closer some of the considerations when we're talking about the pathogens in our gut. I don't think any of you are surprised that a normal, healthy component of our intestines and what's inside of our intestines are things like bacteria, right? I mean, you know this. That's why, a lot of times if your digestion is kind of out of whack, or you're not really feeling good, right? They tell you to take probiotics, right? What are probiotics? Probiotics are just, a lot of the times, just certain types of microbes that are meant to, you eat them, you swallow them, and they replenish maybe the balance of these microbes in your gut. We already discussed from the beginning of the semester this important concept that the good commensal bacteria that exist in the lumen of your small intestines and large intestines. Remember, the lumen just means inside, the space inside of your intestines where all the food is that's being digested and stuff like that, right? The good commensal bacteria that exist in there, right, can become pathogenic if they breach the walls of your intestines and start moving into your tissues. But we said that commensal bacteria serve important beneficial features, right? We need them. We evolved to actually expect these bacteria to be part of us. For example, these are different examples of how commensal gut bacteria help us, right, human hosts. Five different ways that are pictured here. Number one, certain bacteria, like this green bacteria here. So by the way, this white space is the lumen inside of your intestines. And then this is the very delicate, single cell layer wall of your of intestines here, right? And so much of this white space that's not really indicated here would be mucus, right? But many bacteria, like this green one, help us to synthesize or produce certain types of vitamins that our body cannot on our own synthesize, right? So things like vitamin K, right? So certain nutrients are put together by the bacteria to make vitamin K, and then vitamin K is absorbed through the mucosal layer and into our tissues. And vitamin K is very important as a factor in the liver, right? Bacteria help us digest foods. For example, cellulose, right? Our bodies have a very hard time normally on our own breaking down cellulose because we don't actually produce enzymes that are very good at breaking down cellulose. I mean, we break down starch very well, like starch from potatoes, for example, but the cellulose fibers from plants like celery or other types of fruits and vegetables, our enzymes don't do a very good job breaking that down. But the commensal bacteria in our gut, they have the ability to break them down, right? So it can break down plant fibers. I don't know why this cartoon says it breaks into short chain fatty acids, because I think all of you know something like cellulose is a chain of sugars, right? So this could be like individual sugars, right? That's why this is an immunology book, not a cell molecular biology book. But anyhow, let's just go with it, right? The point is that the bacteria breaks these down to small components, and those small components can be absorbed and utilized by your cells as building blocks, right? Importantly, good bacteria can neutralize certain toxic substances. So if you have certain types of toxic substances, obviously bacteria have to be able to deal with toxins as well, right? Because if they're in a puddle somewhere and then this puddle has certain types of poisons in it, these bacteria must be able to deal with those poisons. So they can break down certain types of toxins so that when the pieces of them are absorbed by our gut, they're no longer toxic to us, right? And we said that good bacteria, like the green ones here, their population levels help keep the population levels of bad bacteria, like this red one, in check, right? Because after all, they're all competing for the same resources in there. And therefore, they have to sort of exist in this balance, right? So by having plenty of the green bacteria, you help keep the red bacteria from building up. And lastly, you can see here, the good bacteria, right, are constantly being monitored by the cells of our immune system to make sure that the balance of things like the microbial contents of our gut are normal and healthy. Right? So we will study that a little bit in a few slides. Okay. Let's talk about different opportunities for our immune system to be able to monitor pathogens that are coming in through through our mouth, right? Our gastrointestinal tract that starts with our mouth. So our tonsils, I think we all know that we have tonsils in our mouth. Maybe you don't really know what tonsils really do, right? Why do we have tonsils? Right? When I was a little kid, I actually thought tonsils were the little dumbbell-shaped thing that hangs in the back of your mouth, right? But that's actually your epiglottis. But the tonsils are these things that are sort of these fatty tissue regions that are in the roof of your mouth. So the pharyngeal tonsils are located there, right underneath the layer of or the wall of your mouth, right? Your lingual tonsils. So we have tonsils under our tongue as well. Fatty kind of tissue. And the one I think you're probably familiar with, right, are the palatine tonsils that are on the back corners of our mouth. Because typically these are the ones that we think about, they get infected, right? If you get tonsillitis and then you have to have your tonsils removed. So these tonsils, they are these sort of really fatty, soft tissue deposits, and they represent secondary lymphoid tissues, right? In other words, these are tissues where immune cells go to do their work, right? So these are, look, what are these little sites here? These are germinal centers, right? Very similar to the germinal centers of lymph nodes, right? So this is where things like B cells would activate, be presented with antigen, activate, and potentially, start responding. Tonsils, the way they're also built, because they're soft, sort of fatty tissue, they have a lot of these physical sort of spaces, caverns kind of within the tissue, and these are called crypts. And the purpose of that is to increase the surface area, right? So as you're eating something, right away, your immune system gets a sneak peek at any kind of pathogens that you might be eating with the food, right? So that already your adaptive immune system, for example, right, if it recognizes something that infected you before, right, this is like a heads up that, oh boy, right, what are you doing? You're eating this thing again, right? You're eating this pathogen and it's hitching a ride on the food that you're eating. So your tonsils give your immune system a sort of heads up about that. So let's move farther down into our gut. So in our gut, the mucosal layer that's lined with this layer of mucus, that's what's pictured here, right? This is, let's say, all the way down deep in your gut. We're talking about your small intestines. The mucosa-associated lymphoid tissues that are right beneath your mucosal layers, we call that the MALT, right? Mucosal-associated lymphoid tissues. And this represents specialized secondary lymphoid tissues that essentially surround, right, or are directly underneath that mucosal layer of things like your GI tract or your respiratory tract, or your urinary tract, things like that. If we're talking about our gut, the gut is even more specialized, right? Not only does it have this mucosal associated lymphoid tissue, but it has certain functions that are geared towards dealing with pathogens in our food and things like that. So that surface surrounding our intestines is called the GALT, or gut associated lymphoid tissue. Okay. So in this illustration here, you can see, so this is a section of our small intestine. Here is our thin, delicate, single layer of epithelial cells. And then above that, the gray thing is like our layer of mucus, right? And you can see in the gut lumen, so this white space would be where all your food is passing by and all of the microbes are hanging out there, good microbes that are populating your intestines and normally live there, as well as any microbes that you've eaten that were hitching a ride on the food, right? They're going to come in. And so that's represented by like these little things here, right? These brown sticks are like bacteria. Notice, right, you have immune cells at work here. For example, you have plasma cells that are hanging out right in this GALT layer, this mucosal layer, that are constantly secreting things like IgA, right? And so the IgA antibody, you remember 2 of the Y-shaped IgAs are held together at their stem to make this little bow tie structure. And so then lots of these dimeric IgAs are secreted into the mucus, right? And they sort of are taking part in the mucus to be able to bind to any kinds of antigens that this particular IgA was developed to detect, right? So that's what's shown up here, right? So again, by binding to certain bacteria, you can neutralize the bacteria, you can prevent the bacteria from being able to carry out its normal functions and make it easier for the bacteria to be sort of expelled by your body, right? As they kind of are flushed away by the mucus. You also have dendritic cells. So here's a dendritic cell here. Our friend, the dendritic cell, what do dendritic cells do? Remember, they always want to kind of scavenge around and pick up bits and pieces of interesting antigen in order to show other immune cells, right? Look at what these dendritic cells do, right? They're so eager to kind of sample what's going on in the gut, that they actually kind of reach between the cells of the epithelium. Here's one up here. Look at this, right? It actually reaches between, so it can actually sample, right, some of these microbes that are in your gut. It'll take it in, it'll destroy it, break it down, and then present things like antigenic signals. You also have lots of macrophages, not surprisingly, patrolling around, right? And you have certain memory T cells, right, that are hanging out, right, that maybe your body has selected for previously due to some infection that you had, right? Food poisoning or some other type of situation before involving microbes in your gut causing a problem. I also want you to notice this region over here. You can tell that this seems to be a specialized region, and this is what we call a Peyer's patch. Right? So these are specialized spots in our small intestine. You only have them in your small intestine, where immune cells are concentrated, almost like you're sort of building sort of like a lymph node type of community here, right? And you can see the Peyer's patch is sort of this dome shape, and you have these specialized cells, these M cells, that actually allow samples of microbial antigens and microbes themselves to come in, all right, to be sampled. Okay. Let's take a closer look. Here's a different way of looking at that same picture. You know, I always like showing you at least a couple different ways of looking at things so you don't get stuck thinking that you only recognize a particular cartoon, right? So this is another way of representing what we just talked about. So again, the intestinal lumen in blue here is where the mucus would be. And you have all kinds of dendritic cells here. And you have this specialized Peyer's patch. Right here, you can really appreciate it's like a little dome, and inside of it, you have T cells and B cells that are highly concentrated and ready to go to work. Right? At the top of the dome are the specialized M cells. These M cells are like little sunroofs, if you can think about it that way. Right? They allow antigen and certain microbes that have made their way through the mucus to actually enter. And that's okay, because you might say, well, that's crazy, right? Because I thought the whole point was you're trying to keep microbes out of your tissue, and here you're actually on purpose letting them in, right? Why would you want to do that, right? You want to do that because it's important enough that your immune system is able to monitor sort of like what types of microbes are in your gut, that you're willing to let samples of that come in. But it's okay, because that happens at these specialized Peyer's patches, where you have a ton of adaptive immune cells concentrated here, ready to act, right? So you let little samples of pathogen come in, right? But they're very quickly neutralized and then sort of taken apart. And then your immune system has a chance to kind of look to see what these things are. Right. So not surprisingly, you would have lymph vessels that are draining from the secondary lymphoid tissues, and the purple ones here showing that they are draining to the closest lymph node. Right? So these lymph nodes are essentially just like the lymph nodes that we've studied before, but the ones we've studied before, we are now going to consider those peripheral lymph nodes. Right? So in other words, any lymph node that's sort of not part of your gut or the wall of your gut, so like the ones in your armpit or your throat or whatever, right? Those we just refer to as peripheral lymph nodes, right? They're sort of around the body somewhere. But the ones that are implanted or found right in the mucosal associated lymphoid tissue layer, we will call those the mesenteric lymph nodes, right? So the mesentery refers to a big series of tissue and membranes that hold our guts together, right? So similar type of mechanism, though. You have dendritic cells that were hanging out in the Peyer's patch, and they're bringing samples of antigen into the mesenteric lymph nodes, where you have things like naive CD4 cells that have a chance to wake up, drain out of the efferent lymphatic vessels, and then head to other places in the body. Okay, so here is a electron micrograph high resolution view of one of those little sunroofs of those M cells at the top of the domes. Right? So you can really appreciate, right, that this is like an opening where stuff can go through and get into the Peyer's patch. Here's another view. So bacteria comes in through the M cells of those Peyer patches, where they're very quickly met by things like B cells, T cells, dendritic cells. And so you can see this image on the right maybe is a little bit more a little more realistic looking here, right? So you have all kinds of antigen, potentially bacteria coming in, and they're just met by tons and tons of immune cells, both adaptive immune cells, like things like plasma cells, as well as more innate effectors, things like macrophages, things like dendritic cells, right? And in this way, your immune system is able to constantly sample, right, what's going on inside of the gut. Why? Because think about it, right? The microbial population of our gut is so dynamic because every time you have a meal and eat something, right, there are different microbes that are hitching a ride in, right? So it makes sense that if those microbes are now in your gut and they're finding a very happy place to live inside of your gut, your immune system better keep tabs constantly by sampling what's going on down there, right? So here, another illustration. So M cells allowing things like bacteria or the antigens from bacteria to come into these Peyer's patches where they are met right away, right, by lots and lots of B and T cells, dendritic cells, macrophages, etc. Right? Here again, you have dendritic cells that are actually eagerly sampling the contents of the lumen just by kind of reaching between the epithelial cells. And then you have, you know, regulatory T cells, CD4 cells, all working together. Right? So these Peyer's patches, hopefully, now that I've shown like several slides of them, right, are a unique anatomical feature of our small intestines. And in fact, you might think, oh, we must have lots of these Peyer's patches, right, through our small intestines. In reality, this is a curiosity. We don't. We have something like maybe 100 or so of these Peyer's patches in our small intestine. And they're not spread out evenly. They're actually all sort of congregated or grouped in the last like couple inches of our small intestine before the small intestine connects with the colon or the large intestine, right? So it's just another weird curiosity. I don't know why we evolved that way, right? Because I would have thought, you know, you might as well have these Peyer's patches spread out throughout your entire intestines, right? But for some reason, right, we just have them sort of at the end, and we only have like 100 or so of these little domes to constantly sample things. Maybe it's a way to mitigate risk, right? Because the more of these things you had, the more locations microbes are entering your tissues. Maybe that's just too risky, right? So you have them collected in a certain spot, maybe where you have a better chance of having more immune cells also ready to deal with whatever they're sampling there. Okay. This is a very important concept, right? Because as we're talking about how our immune system has to deal with these mucosal layers differently than it deals with our, for example, systemic layers like our skin. Already we looked at one example, right? Anatomically, it's quite different because whereas our systemic immune system that deals with worrying about things like bacteria coming in through a paper cut in our skin deals with innate effectors that are just kind of hanging out there, but lymph nodes, right? Lymph nodes that are nearby being, or they might not even be that nearby, right? Because if you get a cut on your arm over here, maybe the closest lymph node might actually be under your armpit there, right? So stuff actually has to drain all the way up there before your peripheral or systemic immune system has a chance to kind of like take a look and present stuff to lymphocytes, right? So already anatomically, we said the mucosal layers are different because the mucosal layers, you cannot afford to have to wait to transport something really far away to like some other lymph node in order to check it out. You have to have lots of immune cells already ready surrounding, right, things like your intestines or your airways in your respiratory tract, right? So that's why instead of having reliance on specialized lymph nodes, you can almost think of it as the malt and the galt tissues that surround these different tracts inside of your body are like themselves big spread out lymph nodes, right? Where all these types of antigen presentation and activation of lymph nodes happens in. So anatomically, that's an important difference, right? But also the type of response that your immune cells have to mount in response to something like an infection in your GI tract. Think about, right, if you have a pathogen in your GI tract, it's very easy for it potentially to breach the wall of your mucosal layer. And Therefore, what type of immune response would it need to be met with, right? A big distinguishing factor is whether or not inflammation, right, is a tool used by our immune system to respond. Remember, we said inflammation is very important when we're talking about systemic immune responses, because what does inflammation do? Inflammation, as part of more of an induced innate response, does things like raise the temperature of the skin. It modifies the blood flow in the area to make it so if it swells up a little bit, that's both because you're having leaky blood vessels to allow more complement proteins to get out of your blood into the tissue. You're also, the walls of the capillaries get leaky so that whole cells like monocytes or neutrophils can make their way out of the capillaries much easier, right? And inflammation also results in making it harder for fluid to drain away from that area, right? Fluid that might end up carrying things like bacteria away from the site of where your cut is, right? So therefore, inflammation is a very important part of a systemic immune response. And hopefully, as we've learned about that, right, you're beginning to get a different appreciation of what inflammation really is, right? That inflammation is actually a function of our immune system fighting back, right? However, when you're dealing with the mucosal layers, right, inflammation is really bad, right? Because if you inflame the already super thin and delicate layers of your mucosal layer, what do you risk doing? You actually risk making the breach, right, or the damage to this physical barrier even greater, right? So if you're worried about a few bacteria coming in through a breach in your, let's say, intestinal lining, can you imagine if suddenly the intestinal lining got all inflamed and leaky? You would suddenly have a ton of microbes invading your tissue, right? Therefore, the way our mucosal immunity works is it actually discourages inflammation as part of the immune defense. So that, in large part, is a function of the immune cells behaving a bit differently. Things like macrophages. Because remember, macrophages are one of the first cells in our innate immune system to eat something like a bacteria and send out potentially inflammatory cytokines like interleukins, right? The macrophages that are surrounding our gut mucosa, oh, you better believe there's a lot of them surrounding the gut mucosa, right? But these mucosal macrophages, right, should be a bit different. They don't want to encourage inflammation. So if you look at, for example, a blood monocyte that's flowing through your blood, and this monocyte is going to become a macrophage, right? Eventually, if it exits the blood somewhere in some of your tissues, right, it might become a peripheral systemic macrophage, in which case it's ready to eat bacteria and release interleukins to cause inflammation. But if it exits your blood in your mucosal layers and becomes something like an intestinal macrophage, it's quite different, right? Yeah, I mean, the macrophage can still eat things, phagocytose things, kill things, right? But look at this, right? It loses many of the properties of the systemic macrophages, including things like cytokine production, right? So a macrophage in your mucosal lining will not secrete a lot of inflammatory cytokines. So this is the last slide for today. So this is kind of a complicated slide. I've broken it into two parts, top and bottom. We'll introduce this today, and then we'll elaborate on it a little bit more tomorrow or next time, okay? This top set of panels is what we've learned so far this semester when we're dealing with sort of straight ahead systemic immunity, a cut in your skin, right? So this should all be familiar to you now, right now that we're like, whatever, 8 weeks into the semester, 9 weeks into the semester. So you have your skin, you have some bacteria hanging out on your skin. Underneath it, you have your innate effectors like your macrophages wandering around. Here's MALCOP, right? That's MALCOP wandering around. And then you have a capillary nearby in the wall of the capillary, and this is inside of that blood vessel. So you have things like monocytes, whatever, floating by. If you get a cut, right, the bacteria comes in, macrophages eat the bacteria and release inflammatory cytokines, right? If the macrophages take care of this within a few hours, whatever, that's the end of it, right? But if it turns out that bacteria is harder to deal with or there's too much of it, over time, those inflammatory cytokines cause inflammation. and things like the tissue begins to swell, and you get the leaky blood vessels, right? Leaky blood vessels that allow things like more of these immune cells and antibodies to get into the tissue easier, right? And so that's what really then helps beat the infection, and you're left with, right, some tissue damage that fortunately can just heal up, and maybe you're left with a scab and then a scar, right? But the point is, this is very different if this is happening in your gut, right? In your gut, if you have these bacteria that maybe were commensal, right? Or maybe not, maybe these were bad bacteria, so you had IgAs trying to fight them off. Some of that bacteria might make its way through your mucosal layer and make their way through the lining of your mucosal layer, but they're met by a wide range of different immune cells that are just surrounding the mucosa as part of this malt, right? And you can see right away they go to work to powerfully neutralize the bacteria. And these macrophages that do that don't release cytokines, so that there's no tissue damage above just the damage that was sort of associated with the infection in the first place, right? And as a result, there's no additional risk of more bacteria coming in, and the amount of healing that the tissue has to do is very minimal. Okay. All right. So let's stop there today, and we'll continue this on Friday.